Hello and welcome to the class on Test of Significance, Students T-Test. We studied that the minimum Z values required to reject the null hypothesis are valid only for the cases with large samples. Now, a sample with a size of 30 or more is termed as a large sample. A sample which is having a size less than 30 is described as small sample. And most of the biological experiments involve small samples. The t-distribution enables to make valid conclusions from the small samples. If we plot the frequency curve of the samples of varying sizes, then what we observe is that the amount of space in the tail goes on increasing as the number of observations in the sample goes on decreasing. The t-distribution was proposed by W.S. Gossett. It enables to make reasonably valid inference about the population using the statistic from small samples. The test of significance based on it are referred to as t-tests, and the t-distribution is based on degrees of freedom of a distribution which are identified with the formula small n minus 1. The degrees of freedom of a distribution is defined as the number of variates that can be entered in that distribution before the values of the remainder of variates are fixed by the necessity to produce a certain total. The number of observations which are free to vary will therefore be n minus 1. T-test is used to estimate population mean from the sample mean. It is also known as one-mean t-test, where the population mean is estimated using sample statistic, the sample mean x bar, and population mean is calculated with the formula x bar plus or minus t table value at 5% level of significance and at n-1 degrees of freedom into standard error of the mean. Now when we say that t table value at 5% level of significance and at n-1 degrees of freedom, we need to refer the table of t distribution. Here is an example of estimation of population mean from the sample mean. When the population mean is estimated using the table t value at 5% level of significance, what we observe is that the population mean ranges between 29.7856 to 35.2144, whereas if we are using t-table value at 1% level of significance, what we observe is that the population mean ranges between 28.6 to 36.4. In first case, we are 95% confident about the estimation of population mean, whereas in second case we are 99% confident about the population mean. As the confidence level increases, the precision of the population mean is getting decreased. T-test is also used to test the significance of difference between the means of two small samples which are having different origins, or we can say that the two populations are independent. The independence of the two samples is considered when there is no relationship between the individuals contributing to the sample. The samples are drawn from different populations or from different parts of the same population. In this case, the samples are considered as independent samples. Comparison of such means is known as two-mean t-test, where the null hypothesis is put forth as that there is no significant difference between the sample means. The test statistic is obtained with the formula differences in the means of two samples divided by standard error of the differences between the means. x1 bar minus x2 bar divided by under root sp square divided by n minus 1 plus sp square divided by n2 minus 1. The conclusion is drawn based on comparison of calculated t-value with the table t-value as shown over here. If the calculated t-value is less than table t-value at n-1 degrees of freedom and 5% level of significance, we say that the difference between the two means is non-significant. Null hypothesis is therefore failed to be rejected. If calculated t-value is greater than the table t-value at n-1 degrees of freedom and at 5% level of significance, we say that the difference between the two means is significant. Null hypothesis is rejected. If the calculated t-value exceeds the table t-value at 1% level of significance and at n-1 degrees of freedom, we say that the difference between the two means is highly significant. Null hypothesis is rejected in this case. T-test is also conducted to test the significance of difference between the means of two small samples and they are dependent on one another. That is to say that the samples are drawn from the same population. This is also known as a paired t-test. Two samples are dependent when they have some common factors linking the observations in the two samples. For example, the values are obtained before and after the treatment, or the values obtained after controlled treatment and experimental treatments, or the values obtained at two different periods. The significance of mean difference is tested using paired t-test. Here, the first step is to put forth the null hypothesis that there is no significant difference between the sample mean difference and the population mean difference, and the value of t is obtained with the formula sample means difference minus population means difference divided by standard error of the mean difference, such as t is equal to capital D bar minus mu d divided by standard deviation capital D divided by under root small n minus 1. In this case, there are some basic computations which are required to be performed before the test, which are shown over here. Again, here in this particular case, the decision will be based on comparison of the calculated t-value with the table t-value as shown over here in this slide. The t-test is even applied for testing the significance of the coefficient of correlation. The observed coefficient of correlation is significant or not can be tested using a sampling distribution of sample correlation coefficients if it is a large sample, and using t-distribution if it is a small sample. The standard error of correlation coefficient for large samples is computed using the given formula, whereas the standard error of the correlation coefficient for small samples is computed using the given formula. Where, for example, if in the given data small n denotes the number of pairs of the data, small r denotes Karl Pearson's coefficient of correlation, then we can place the null hypothesis as r minus rho is equal to 0, where rho denotes population correlation coefficient. The value of z or t is obtained according to the number of observations in the given sample using the given formula. The table z or table t at specific level of significance and at n-2 degrees of freedom is referred, and the decision will be based on comparison of calculated z or t value with the table t or z value. Thank you.